Let's jump right in. Welcome to this explainer on ATP synthesis. So, if you think of ATP as, the universal currency of the cell, then mastering how it's made is literally like knowing how to print your own money on the Biology 9700 exam. These concepts, they're fundamental, they're everywhere, and they are incredibly valuable. But here's the catch. The difference between getting a B and scoring that A star usually comes down to one tiny costly mistake. Today, we aren't just reviewing the pathways. We're going to look at this squarely through the eyes of a senior examiner to uncover exactly where students are unknowingly leaving guaranteed marks on the table. We're going to master the underlying biology and then, more importantly, master how to express it perfectly. And look at this quote straight from the principal examiner report. Candidates consistently confuse key terminology and cause and effect relationships. Examiners see this specific pattern year after year. Honestly, you could spend weeks memorizing the entire textbook chapter on ATP synthesis, but if you mix up the engine with the fuel, like if you can't articulate exactly what biological event drives the next one, those top bands are going to stay completely out of reach. It's the exact reason why brilliant students sometimes walk out of the exam room feeling totally confident, only to be totally shocked by their results later. So let's clear up that exact confusion right now, starting with the big one. First up is chemiosmosis. Now, don't let the long, intimidating words scare you off. Just think of it like a massive hydroelectric dam. Chemiosmosis is simply the synthesis of ATP driven by a flow of protons across a membrane, moving down their electrochemical gradient. If you write something super simple on your exam, like hydrogen ions move, you are completely missing the dam and the water pressure. You're basically making the examiner guess that you know the underlying principles. You absolutely must specify the membrane, which acts as the physical barrier, and the electrochemical gradient, which acts as the pressure. Without those two crucial components, well, the ATP synthase enzyme has literally no power to spin and create that cellular currency. Okay, now contrast that massive complex dam machinery with a substrate-linked reaction. This is a much more direct approach. It's kind of like handing someone cash instead of setting up a complicated bank transfer. It's just the direct transfer of a phosphate group straight from a substrate molecule directly to ADP. This forms ATP entirely without that proton gradient we just talked about. Top-performing students can instantly tell you whether a process is using the complex membrane-bound gradient of chemiosmosis or the direct enzyme-catalyzed handoff of a substrate-linked reaction. Knowing this fundamental difference, absolutely non-negotiable for tackling those advanced pathway questions. So how do we actually turn these essential biological definitions into an A star? Well, we use a highly reliable framework. Problem-solving in multi-step biological pathways requires a really systematic approach. First, you deconstruct. You isolate the given data from the prompt. Next, you solve, breaking the biological pathway into logical, sequential steps without skipping ahead. And finally, you evaluate. Check if your biological conclusion actually makes sense in reality. I know, under exam pressure, it's incredibly tempting to panic and just word vomit everything you know onto the page all at once. But taking a breath and deliberately applying this three-step structured approach, it changes everything about how your answer is graded. In fact, this framework is your ultimate safety net, thanks to something called the method mark strategy. Here's a little secret for you. Examiners actually want to give you points. If you lay out your working clearly, identifying the key elements and showing clear cause and effect reasoning, you can actually earn marks for your scientific process, even if your final conclusion goes slightly off track. You are literally getting paid in marks just for showing you know how to think systematically like a scientist, step by careful step. Now, there is a hidden clue in these exams that completely flips how you should format your answer. I'll reveal what that is in just a moment because once you see it, you honestly can't unsee it. But first, we need to navigate the three big traps that make this clue completely invisible to most test takers. Because if you fall into these common traps, even the most perfect conceptual understanding of the biology won't save your grade. Right, so principal examiner reports explicitly warn us about three fatal mistakes. Number one, confusing cause and effect. Number two, forgetting to check units and terminology. And number three, giving answers that lack specific biological context. It's an absolute tragedy when a candidate perfectly describes the general mechanism of ATP production, but completely ignores that the question is specifically about, say, a plant cell operating in anaerobic conditions. The exam isn't testing if you memorize the textbook. No, it's testing if you can apply those concepts to the exact, unfamiliar cellular environment they just handed you. Context is absolutely everything. Which brings us to a massive examiner alert. Always define your key terms before explaining the process. If you just start listing facts without establishing your foundational elements first, your argument just collapses. Actually, a really great hack here is to use a mnemonic to remember the sequential steps of ATP synthesis. Think of something silly like, awesome protons make tremendous energy. Whatever weird phrase sticks in your brain. It ensures you never skip building that solid foundation before you start constructing your complex, extended response under extreme time pressure. Let's escalate this to experimental data, because your assessment objectives heavily target your ability to handle evidence. You must know your statistical matrix inside and out. So if you're comparing the mean ATP yield of two entirely different cell types, that's a t-test. Looking at categorical data, like the strict presence or absence of a specific respiratory enzyme, that is chi-squared. Or maybe you're tracking the rate of ATP production as the environmental temperature steadily rises. Well, that's a correlation test. Choosing the wrong tool doesn't just look bad, it completely invalidates your entire analytical process in the eyes of the examiner. Let's put your examiner hat on for a second. Think about everything we've just covered regarding biological context, causal relationships, and systematic analysis. Pause for a second when we look at this next scenario. I want to see if you can spot the trap that caught thousands of other students. Let's stack a typical, flawed student answer right up against a top-tier mark-maximizing one. Notice the striking difference here? The flawed answer just violently dumps memorized facts onto the page, hoping something, anything, sticks to the mark scheme. But the top-band answer? It builds a bridge, it applies our deconstruct, solve, and evaluate framework directly to the exact scenario. The top-scoring candidate acts like an active research scientist. They question limitations, they consider alternative interpretations, and they justify every single biological decision with hard evidence extracted right from the prompt. They don't just recite, they evaluate. To reliably hit that top band over and over again, your brain needs to automatically process every complex question through a strict 4-step sequence. Step 1, state the core theoretical concept clearly. Step 2, back it up with the specific data or evidence provided in the prompt. Step 3, connect the two using logical, causal, scientific reasoning. And step 4, ensure your vocabulary strictly matches the precise terminology examiners demand. It's a chain, right? If you skip a step, the logical chain breaks, and those marks drop instantly. And this high level of reasoning means you absolutely cannot study ATP synthesis in a vacuum. The conceptual framework of Biology 9700 is deeply interconnected. For example, when you can conceptually connect the membrane transport principles you learned way back in Chapter 4 directly to the intricate proton gradients of chemiosmosis we discussed earlier, examiners really notice that. Referencing these cross-topic connections demonstrates a master-level, holistic understanding of biology, and as the report says, that achieves significantly higher marks every single time. But here is the ultimate twist, and it is a painful one. You could have absolutely perfect biological knowledge. You could have flawless statistical analysis. You could outline the most beautiful, nuanced mechanism of chemiosmosis the examiner has ever seen. But what happens if you answer the entirely wrong question? Unfortunately, you will score a 0 for that section. The hidden cause of missing those top mark bands almost always comes down to misunderstanding the command words. This is that hidden examiner clue I promised you. You might be perfectly describing a beautiful pathway when the examiner explicitly asked you to explain why it happens. Those are two completely different tasks. So the next time you sit down to tackle a complex biological pathway, I want you to ask yourself, are you just proving what you know, or are you actually answering what they asked? Because the difference between those two approaches, that is exactly the difference between a B and an A star.